Good morning, Melania Cristescu. Welcome on VH Berries.
Thank you, Victor, for having me. It's a pleasure.
So we are in your home in Montreal. You are a teacher in biology at McGill. How are you doing these days?
I'm doing very well. Thank you very much. I am a professor at McGill University in the biology department, and I work in an area called Ecological Genomics. So I'm using genetic tools, like genes, to answer broad ecological or evolutionary questions.
I have so much respect, and I'm so grateful to have this opportunity to talk to you and your work, because you are studying very small things, taking some really small human beings from Asian lakes, for example.
Yeah, I'm working with aquatic organisms. So you are correct. I'm working in aquatic ecosystems, generally freshwater ecosystems. And I look at how aquatic organisms evolve or speciate, and I also look at the human impact on those organisms.
And it all started a long time ago in Romania, in Europe.
Indeed, I had an interest, actually a dream of becoming a marine biologist when I was very young. And I grew up in Romania, central of Romania, a region called Transylvania. And I was introduced to nature by my father. So I used to spend lots of vacations in nature, exploring places, and at the Black Sea coast.
Exploring at the Black Sea coast. Is it a special region in Romania?
Well, I did my undergraduate degree in Constanța, a city that is located at the coast of the Black Sea. So I had the chance to work on aquatic systems since I was an undergraduate student, and I very much enjoyed exploring the sea.
This is interesting because now I can see a connection, because maybe that in Romania, in your hometown, there must be a lot of lakes, a lot of nature. So,
well, I was born in the mountains with a love for water.
So since then you decided to move in Canada. Tell me about your journey.
Well, it was not a very straightforward journey. I wanted to study in Canada, so I came to Canada in 2000 to join graduate school. First, I landed in British Columbia, and after about a year, I moved to Ontario, and I follow undergraduate program at University of Guelph. I was very lucky to work with a very dynamic research group and to meet exceptional people that introduced me to molecular methods in biology. Many aspects were totally new to me.
That's interesting, because in science, mentors plays a huge role, because you are right now inspiring a new graduate student, but in the past you were also influenced by them.
That's true. It is a multi-generational process. So I was lucky to have amazing role models, and I'm trying to take aspects of their teaching and transfer that enthusiasm to the next generation. You are right. And that is a lot of fun.
So, Melania, in McGill, you have a lab called the Cristescu Lab, in which you are doing experiences and...
Yes, in our lab, we use a combination of approaches. We use experiments in lab experiments, but we also work outside in nature. We have also a large experimental field station at Mont Saint-Hilaire, where we have 100 artificial ponds in which we experiment with our organisms, aquatic invertebrates. So, and we do at the same time molecular work. So our work is very much interdisciplinary. So we work with large teams that bring into the play different expertises.
And I assume that while you are working and doing your work, Melania, you are facing sometimes some failures because in science, this is the key to find new solution.
You're correct, and that is one aspect that requires a lot of patience and training of graduate students. I think it is hard for young researchers to deal with failures and to accept that this is part of the road, that almost no experiment works the first time you attempt it. And you have to explain that is normal, and what would be unusual will be to have that perfect result from the first time you set up an experiment.
For example, with Edison, with his 10,000 tries before inventing the lights.
Yeah, well, we don't have as much time, but in a graduate program for large-scale experiments, often we do 2-3 trials until we get good results.
And for the last, let's say, 10 years, what discovery have you made in this Cristescu lab?
Wow, that is a hard question to answer because we work on different fronts. So one line of research looks at how mutations accumulate in genomes, so in the population of organisms, and how these mutations accumulate under severe environmental stress. For example, in habitats in which we have pollution, metal pollution, for example, in lakes contaminated with nickel or copper. We chose these particular stressors because the Sudbury region on Ontario has been highly affected by pollution. So I have an experiment that we started more than 10 years ago, in which we propagate organisms under exposure of nickel and copper, and we monitor through time how mutations accumulate in this genome. And we also try to monitor what is the impact of these mutations in the fitness of the organism, in the well-being of the organism, the health. And we were lucky to come across very unexpected patterns. For example, one important result in this line of research was that most of the mutations that we capture are not the typical point mutations or short mutations. You can imagine the change of a letter in the genetic code. Instead, you have large-scale deletions and duplications, so major changes in the genome. You can imagine changes, let's say a whole page of the genetic story is being replaced or duplicated. So we also noticed aspects of recombination, large-scale recombinations that happen in organisms that do not reproduce sexually. So we thought before that asexual organisms transfer these genomes from generation to generation in unchanged, almost like a frozen genome, in a clonal propagation. And we found that that is not the case, that there is a lot of change that happens in these genomes, even if the organism reproduces clonally.
I understand, and you can often compare the DNA that you are studying with some barcodes that we can find on the product of the supermarket.
Well, that is another line of research that we are doing in the lab, where we try to use the genes, the molecular tools, to identify species. And the reason this is extremely important is because we do not have the expertise to use morphology to identify species. So it is extremely hard to quantify biodiversity and also to quantify biodiversity changes. And as you know, humans have a dramatic impact on biodiversity these days. And in order to monitor, for example, the impact of global change on particular ecosystems, we'll have to have very efficient tools to monitor biodiversity and these changes. So because of this limitation, we are employing now these small genes, small fragments of genes in order to discriminate different species. And we do that in the same way products in a market are being labeled with a barcode. So you can imagine these genes being equivalent with the barcode of a product, because the fragment of these genes is unique to each species.
You mentioned the word ecosystem, and I assume that during the last decades, you saw the impact of climate change on lakes, on the forest, on mountains.
We don't necessarily do long-term monitoring to follow, to trace very closely these changes. We are more interested in developing these efficient tools and provide those tools to agencies, to governmental labs, or other labs that are interested in monitoring these changes. So my lab is more at the forefront of developing these methods and advancing these methods, especially the very non-invasive and sensitive methods that are based on environmental DNA and environmental RNA. And the reason we do that is because we want to provide tools that allow a researcher to describe all the species in an aquatic habitat without sampling the species, with just collecting the DNA that leaks from the organisms into the habitat. So it's in a totally non-invasive way.
I believe it always starts with a specific approach. So I'm very curious, Melania, about the starting point that you are choosing every time that you are beginning a new research.
Well, selecting a particular project and formulating a research question or formulating a hypothesis that has very clear predictions is the core of doing science. And you are correct. This is perhaps the most important part of the research. Selecting well the problem that you want to tackle and formulating very well the research questions. And often graduate students work for their first year on that hypothesis, formulating that hypothesis very clearly, and also being able to come up with very clear predictions that could be proved or disproved.
So you've been interested in the very small, but are you interested in space, in things that sometimes can be bigger than us?
So this is a very interesting question. Of course, I've been always fascinated about thinking about the extraterrestrial life, how life might evolve on other planets, other solar systems, other galaxies. I tend to believe that there must be life outside the blue planet. It is hard to imagine what form of life that would be, but I believe that any environment that holds water in a liquid form will be prone to the evolution of life. And it is also very possible that the molecular form, that's, let's say, life as we know in cellular form, with a genetic code based on DNA and with a messenger called RNA, would be at the base of pretty much any life in the universe that is based on carbon and water.
That's why I believe that in your work there is math, science, but there is so much philosophy, because there are questions that cannot be solved. And I also believe that our knowledge, the human knowledge, is at like less than 1%. There is so much things that we don't know yet.
Well, this is why it's fascinating to work in science, because each question that we answer opens up many more questions. So we never actually run out of questions. So the more we understand of a process, the more it opens up for more understanding. So it's almost counterintuitive.
Yes, because the more you are discovering things, you realize then that there is a new field of possibilities, and that it will never stop.
That's true. There are aspects that we can describe to a level of depth that is very comprehensive, and it can be considered an end product. But most of the areas of explorations are just the beginning.
I've read a lot of books written by Yuval Noah Harari. He's talking a lot about the past and the future, and I assume that you are also interested in both, because you are trying to understand the problem of the past. For example, some viruses that can affect the human species, to better prevent them in the future.
Well, I'm at the core an evolutionary biologist, so I'm thinking about my problems in an evolutionary context. I'm trying, when I want to know how a species might evolve in the future, we have to look in the past to see how that particular species adapted in the past to current environmental conditions. And the genomic area, or the genomic research, is extremely intriguing, because it does allow us to make these trips back in time. So you can imagine that genomics is like a time machine that allows you to travel back in time to figure out the most important evolutionary events that led to the diversification of life now on the planet. So it allows us to figure out what were those genetic changes that allowed for these revolutions in the way species evolved. Like these very spectacular evolutionary transitions and adaptations.
At McGill University, you are doing more research than teaching now.
That is correct. I do a bit more research than teaching, but I'm also involved in teaching undergraduate students, first-year biology students, in organismal biology, an introduction in organismal biology. So I teach them about the diversity of animals, and I focus on invertebrates because they are very, very abundant. So 97% of all animals are actually invertebrates. So that is what I'm teaching.
So you are taking the subject that are the most unexplored.
And bring it to their attention, and with the hope that they are going to appreciate the diversity of life and how little we actually know about the diversity of life. And also, I try to make them aware of the biodiversity crisis that we are experiencing. So one of the most research directions that we are taking involves the use of environmental RNA and DNA that I mentioned. So these molecules that are being released by organisms into the system. And to give you an example, we can take a water sample. So like a small half a liter of water from a lake, we can extract the DNA that is present in that water, and we can amplify targeted places in the genome, and based on that information, using next generation sequencing, we can describe all organisms that live in that habitat, from bacteria to fish. And we can do that in a very non-invasive way, in a very sensitive way, because often it is very difficult to capture all organisms, to see them. Let's say, for example, to fish species that are rare. So this method allows us to describe biodiversity in a very sensitive way and in a very non-invasive way.
And I believe that during all the year, the 4 seasons in Canada, there is some seasons, for example, in winter, in which there is more specific things to do because there is a special weather and with the snow and ice.
Well, we do try to sample our habitats across the season. For example, when we want to follow seasonal changes, or sometimes particular environmental factors can influence the sensitivity of our method, can influence, for example, the way DNA molecules preserve in a habitat. So we work under different conditions and in different seasons.
That's also can be linked with agriculture. So does your studies, for example, can cover some vegetable, some fruits? Because I believe that they also have inside them some DNA.
Well, I don't work directly, with plants, although the methods that we develop based on environmental DNA can be applied to terrestrial environments and can be applied in agricultural related questions. For example, one important aspect in agriculture is identifying pathogens. So, we also look at the impact agricultural practices could have on aquatic ecosystems, because often nutrients or pesticides leak from an agricultural land into the watershed and is being then released into the Great Lakes and eventually into the oceans. So generating a cascade effect sometimes at a very distant location.
And next to your job as a scientist and teacher, when you're doing some hiking in the mountains or in Canada, do you always think about it when you're seeing some lakes and some mountains?
That is true. It is very tempting. When I drive around, I have in the back of my car, in the trunk, always my sampling gear.
Whoa.
So if I see a habitat, I stop by and I sample it. It is very tempting to do that.
Sure. So you're bringing them in the Cristescu lab to
analyze the sample. Yeah, I do that, or even for fun, if I'm with my children to teach them a particular technique of sampling, if I'm with my students to show them particular aspects of a habitat. It is fun to drive around and explore places.
And right now on websites such as YouTube, we see a lot of channels that are covering science, that are trying to simplify everything so the mainstream audience can understand it. Does it interest you? Do you have a special look toward this content?
Well, this is indeed extremely challenging for us to bring complex research or complex phenomena to the broad audience and to bring it in a way that not only that makes sense, but also captivates the audience, inspires, and makes a difference. So we tend to train students, graduate students, mainly into the research writing and on presenting their results to the scientific community. And I think we don't do a good enough job into promoting the research to the broad audience. And I think this is a mission that universities should be embracing more.
You're telling me, Melania, that you're going to start a podcast or a video program soon.
That would be good, yes. I don't think I will do it, but I think we need, as a scientific community, to do that more often. So this is why I'm excited to be here today with you.
Thank you so much, Melania, for your time. You are such an inspiring person. Thank you so much.
Thank you, Victor. It was a pleasure talking to you.